Hello friends, first of all thanks for being here in my YouTube channel. Today we are going to see the diseases of Brinjal. First disease, that is little leaf of Brinjal, which is caused by the phytoplasma, means the causal organism, that is phytoplasma. Let me see the symptoms. Look at the title of the disease, that is little leaf. Here some points can be clear very easily regarding the symptoms. That is, the disease is exhibiting the symptoms regarding the leaf, means that is the little leaf. The small or little leaves, that's why it is called as little leaf of Brinjal. It is the characteristic symptom. Characteristic symptom is what? That is only particularly related to that one disease. Other symptoms are very common which can be seen in each and every diseases. For example, drying, fruit development, then stunting of growth, etc. The petioles are so short that the leaves appear to be sticking to the stem. The affected plants have narrow, soft, smooth and yellow shorter leaves. Means near about all the symptoms are related to the leaf and its development. Newly formed leaves are much shorter. Internodes of the stem are also shortened. Auxiliary buds get enlarged, but their petioles and leaves also remain shortened, giving the plant a bushy appearance. Means the bunch-like structure of that leaves you can observe very clearly the plant which is affected by the phytoplasmas. Mostly there is no flowering, but if the flowering is there, then they are formed remain green. Fruiting is rare. If there is flowering is affected, then if there is no flowering, then there will be no chance of getting the fruits. If the flowering is affected, then fruits will also get affected. The fruiting is rare. If any fruit is formed, it becomes hard, tough and fails to mature. Young fruit turns necrotic, get mummified and cling to the plant. See here, young fruit turns necrotic. What is meant by necrotic? Then necrosis is nothing but the death of the tissues. This situation or this condition is called as necrosis or necrotic lesions. If the part of the plant of leaf showing the death of particular tissues, then there will be some spots may occur. That lesion is called as necrotic lesions. You can see in this picture the little leaves of Brinjal exhibiting the bushy appearance. Let me discuss about the pathogen. It is also called as etiology, means study of that pathogen, means how does it appears, how does it look like, means whether it is septate, aseptate, hyaline, conidia, etc. The disease is caused by phytoplasma having ovoid to spherical body and which is concentrated in the phloem sieve tubes. Okay, this is all about the pathogen. Let me discuss about the spread, that is survival and spread. It is also called as perpetuation in the presence of main host or in the absence of main host, on the collateral or on the alternate host. How does it survive? The pathogen perinates in weed host and jacid. The disease is also transmitted by Emposca devastans. However, it is a less efficient vector. The variety Pusa purple long and selection T are highly susceptible. Farmers should avoid such kind of varieties for cultivation or for sowing. Collateral host Datura fastuosa, this will act as a collateral host for this phytoplasma. Next point, that is the management of this pathogen. First of all, use tolerant varieties. For example, Pusa purple round, Pusa purple cluster and Arca shield. These are the tolerant varieties. They can withstand even there is an infection, there is an incidence of phytoplasma. Then also the plant will not exhibit any symptoms or will not show any reduction in its production of fruits. Next point, destruction of affected plants means if you observe any infected plants or affected plants by the phytoplasma, suddenly such kind of plants should be rogue out from the plot. Eradication of solanaceous weed host means the weeds which are belongs to the solanaceous family, that should be removed from the growing area. Spray methyl dimeton 2 ml per liter or malathion 3 ml per liter, or soil application of borate granules. So let me discuss about the next disease of brinjal, that is bacterial wilt. Bacterial wilt of brinjal, it is caused by Ralstonia solanacearum. It is the causal organism. Symptoms: sudden wilting and death of infected plants is the characteristic symptom. As we know, the wilt, it is the symptom, means the situation in which the infected plant shows drying and ultimately death. It may be complete death of plant or partial death of plant parts, and such kind of situation is called as wilt. And here this wilt is caused by the bacteria, and the first symptom we have seen, that is the sudden wilting of death and death of infected plant is the characteristic symptom. The next symptom, the petiole of older leaves droop down and the leaves show epinasty symptoms accompanied by yellowing and stunting of whole plant. Look at the second symptom, that is the stunting of plant, means there is no further development of plant occurs. There is the disturbance of flowering, there is the disturbance of proper growth of the plant parts, there is the disturbance in the fruit setting due to the infection of Ralstonia solanacearum, which exhibit the symptoms of wilting. Look at this picture. You can partially observe the plant showing the partial wilt situation. Next symptom, typical browning of vascular tissue of roots and stem can be seen. Means brown colored vascular tissue can be observed both in roots and stems. From cross sections of infected plants, whitish bacterial exudate comes out. Means there is the presence of bacteria you can observe. Let me discuss regarding the pathogen. The bacterium is gram-negative, motile, rod-shaped, having rounded ends with 1 to 4 polar flagella. We know the role of flagella, and there is different flagellar arrangement in case of bacteria. Race 1 of R. solanacearum affects solanaceous plants such as tomato, eggplant, and may other non-solanaceous plants also. Mode of survival and spread, that is perpetuation. The bacterium is both soil and seed-borne in nature and overwinters in infected plant parts, in wild host plant and weeds. Spread through irrigation water or infested soil and agricultural implements. Favorable conditions. Relatively high soil moisture and soil temperature favors the disease development. This is very important factor. Climatic situation or climatic conditions plays an important role for the incidence as well as the development of the disease. Next, management. Grow resistant varieties like Pant Samrat, Arkanidhi, Arka Keshav, Arka Neelakantha, Surya, etc. Crop rotation with non-solanaceous host. Farmers should avoid growing of same crop year after year or season after season on the same piece of land. That is, avoid monoculture or monocropping pattern. Green manuring with brassica species. Soil solarization with transparent polythene sheet. Next, that is the biological control with Pseudomonas fluorescens, etc. Next disease of brinjal, that is Phomopsis fruit rot or blight. This is very important disease as it causes a great loss to the plants and ultimately farmers. The causal organism for this, that is Phomopsis vexans. The disease is severe in tropical and subtropical areas of the world. In India, it was reported in 1935 in Gujarat. The pathogen attacks foliage and fruits, but the latter phase is more destructive. Let me see symptoms. The plants are attacked at all stages of growth, producing damping of symptoms in nurseries and color rot on young plants, on leaves, circular to irregular, clearly defined grayish brown spots having light centers appear. The deceased leaves become yellowish in color and may droop off. Several black pycnidia can be seen on older spots. Next symptom, the lesion on stem are dark brown, round to oval and have greyish centers where pycnidia develop. At the base of the stem, the fungus causes characteristic constrictions leading to canker development and toppling of plants. On fruits, small pale sunken spot appear which on enlargement cover entire fruit surface. These all symptoms you can observe in these two pictures. Next symptom, these spots become watery leading to the soft rot phase of the disease. A large number of dot like pycnidia also develop on such spots. The infection of fruit through calyx leads to development of dry rot and fruit appear. Let me discuss about the pathogen. The mycelium of the pathogen is septate and hyaline, becoming dark with age. Pycnidia are submerged and later become erupt with a prominent ostiole. Conidia are produced on simple to branched conidiophores and are of two types, alpha conidia which are subcylindrical and beta conidia which are filiform and curved. Role of beta conidia is the epidemiology of the disease is not very clear. The perfect stage produces perithecia in which asci with 8 hyaline, bicelled, ellipsoid, fusoid ascospores are produced which are usually constricted at septum. Perfect stage means where sexual stage is present, that is called perfect stage. Mode of survival and spread, that is perpetuation. Primary infection, pathogen is seed borne and also survive in plant debris as mycelium and pycnidia. Secondary infection, conidia dispersed through rain splashes, irrigation, agricultural tools and insects. The favorable conditions for the disease development are highly relative humidity coupled with higher temperature favors the disease development. Maximum disease development takes place at about 26 degrees Celsius under wet weather conditions with 55% relative humidity. Ultimately, we have to manage this one disease by adopting the different management practices that are as follows. Removal and destruction of diseased crop debris, practicing crop rotation. As I told you earlier, one should avoid growing of same crop or single crop year after year or season after season on the same piece of land. They can avoid monoculture and adopt crop rotation. The summer plowing helps in reducing initium inoculum. Means if the farmer adopt the deep plowing, the deeply seated inoculums in the soil may get exposed to the sunlight and that may be killed by the sun rays. Use of disease free seed. Hot water treatment of seed at 50 degrees Celsius for 30 minutes. Seed treatment with thiophanate methyl at 1 gram per kg seed. Spray twice with thiophanate methyl or carbendazim at the rate 0.1% at 20 days interval. This is the very effective management practice. Thank you.